Hello everyone, my name is Shadi Einollahi and I'm going to present one of my research studies on the comparison of uric acid enzyme production in clinical and environmental strains of Pseudomonas and Bacillus. Let's get started by a brief introduction of the uric acid enzyme. This enzyme is also recognized as urate oxidase or urate oxidoreductase, and it's the enzyme that catalyzes the oxidation of uric acid to allantoin and for sure plays an important part in nitrogen metabolism. There are a number of microorganisms able to produce uric acid. However, for the current study, we will be focusing on Pseudomonas aeruginosa and Bacillus cereus. Now I would like to attract your attention to the aims and objectives of the study. First of all, I will be speaking about the isolation of uric acid producing strains of clinical Pseudomonas and environmental Bacillus. Then I will move on to the identification of strains with the highest uricolytic activity. Next, enzyme production and uric acid assay will be focused on, and various parameters to optimize the production of the enzyme will be discussed. Finally, I will conclude by enzyme purification. Well, I gathered about 20 strains of Pseudomonas aeruginosa from a hospital and then cultured them on nutrient agar supplemented with uric acid. The uric acid producing strains were isolated based on uricolytic activity on agar plate, which was identified as zone of clearance around the colonies. The strain with the highest uricolytic activity was confirmed by standard biochemical tests, and then I cultured the strain in production medium. I also isolated uric acid producing Bacillus strains from active sludge using pour plate technique in basal salt medium supplemented with uric acid, which were identified as Bacillus cereus based on standard biochemical tests, and then I cultured them in the production medium. After I cultured the selected strains in the production medium, the medium was incubated and then centrifuged to be ready for the uric acid assay using a spectrophotometer. Then I tried to maximize uric acid production by considering parameters such as uric acid concentration, temperature, pH, carbon source, and incubation period. And as you can see on this slide, the graphs illustrate the optimized uric acid production. As you can see on this slide, after I inoculated the bacteria in the optimized production media, uric acid enzymes produced by both Pseudomonas aeruginosa and Bacillus cereus were concentrated and purified by using ammonium sulfate precipitation and dialysis. And finally, uric acid production and the level of purification were confirmed and assessed by polyacrylamide gel electrophoresis and Bradford protein assay and by drawing a standard curve for bovine serum albumin. Also, the tables on the following slides compare data on enzyme activity, total units, protein content, total protein, and specific activity for the produced enzymes by both Pseudomonas aeruginosa and Bacillus cereus at different stages of enzyme purification. On this slide, you can also see the standard curve for bovine serum albumin for the calculation of protein content by Bradford protein assay. Now here you can see the estimated weights of the produced enzymes by SDS page. I will just conclude by drawing your attention to the following suggestions for future research. First of all, molecular identification of the uricase producing strains along with identifying the gene encoding uricase enzyme are highly recommended. Moreover, recombinant DNA technology and genetic engineering can be applied to clone the gene encoding uricase enzyme in non-pathogenic bacteria. Finally, the enzyme can be purified completely by using chromatography techniques. Thank you very much indeed for your time and attention. Goodbye.